Hello students, today's class is on wet mounts and staining methods used in microbiology, particularly in bacteriology. Learning objectives for today's classes: at the end of the session, the nursing students should be able to know types of specimen preparation, method of specimen preparation, and bacterial staining methods. So if you look into specimen preparation, there are mainly two types of specimen preparation. One is wet mount preparation and other is fixation of the specimen and staining. Wet mount preparations are those wherein the clinical sample you receive are just mounted onto the glass slide. A cover slip is placed on them and they are observed on them under the microscope. So here you can use either a drop of saline along with the specimen, or sometimes you can use certain stains. So the clinical specimens which are prepared as wet mount are urine specimen, stool, and certain skin scrapings, hair and nail. Looking to fixation and staining of the specimens, here the specimen is fixed onto the glass slide. A smear of the specimen is prepared on the glass slide. It is fixed either by the heat or the chemical method, followed by staining. The specimens which are subjected to fixation and staining are sputum sample, pus sample, and body fluids like cerebrospinal fluid you obtain from the brain and other body fluids. Further looking into types of wet mounts, they can be live mounts, that is those wherein you can see the live microbes, like saline wet mount, and a variant of the saline wet mount is a hanging drop wherein you can demonstrate the motility of the bacteria. KOH mount, particularly useful for fungi, and stained mount. These are the mounts wherein you add in a drop of stain like iodine. Iodine mounts are particularly useful to observe parasites like protozoa. And LPCB mount, which is lactophenol cotton blue mount, particularly useful for studying the morphology of fungi. Looking into fixation and staining, there are certain stains like simple stain, differential stain, and special stains used in bacteriology. So first let us look into the bacterial cell, how it looks. So I hope you remember how the bacterial cell looks. The outer blue one is the capsule. Here it is highlighted in red if you can see. It is not present in all the bacteria, but couple of bacteria have a capsule and outer coating. The main outer layer of the bacterial cell is the cell wall. Please remember this. And then internally there is a cytoplasm which is bounded by the cell membrane or the plasma membrane. And within the cytoplasm is the DNA, which is ill-defined, arranged as a nucleoid. And there are certain ribosomes within the cytoplasm. Apart from this, some bacteria have flagella. You can see the pink extensions which are flagella, which are organs of motility, which are present only in, not in all bacteria, but in some bacteria. So you have to remember that there's a cell wall, the cytoplasm. Within the cytoplasm, there's an ill-defined nucleus and ribosomes, and the cytoplasm is bound by the cell membrane or the plasma membrane. And only some bacteria have an outer coating called as capsule, and a couple of them have flagella. Okay, not all bacteria. Now let us know the specimen preparation methods. Looking into wet mount, we have already studied this is saline wet mount, wherein we can detect bacterial motility by hanging drop method. And other use of saline wet mount is detection of trophozoites and cysts of protozoa and eggs and larval forms of helminths. KOH mount is a potassium hydroxide mount used to study the morphology of fungi. Lactophenol cotton blue mount is also used to study the morphology of fungi, and iodine mount is particularly useful to identify the ova and cysts of the parasites. So these are the images showing the wet or the saline mount. They're showing the cysts of the protozoa, the first image. The next one is the potassium hydroxide mount, which is showing the hyphal forms of the fungi. Below two are the stained mounts, that is lactophenol cotton blue mount, where you can see the fungi which are stained dark blue in color, and iodine mount, wherein you can see the eggs of a parasite known as trichuris, which are stained golden yellow in color. So which do you feel is better? Is it a plain saline mount or a stained mount? Definitely a stained mounts are better. They show the better morphology of the microbe. So what are the requirements when you prepare a wet mount? What are the materials needed? So first one is the microscopic slide. Okay, to observe under microscope, you definitely need to mount the specimen on the slide. So then we need some cover slips. Once we have mounted the specimen or put a drop of specimen on the slide, we need a cover slip to cover it. And then how do we put this specimen on the slide? By using an inoculating loop, which is shown below. This is one of the material used. And next, to place the cover slip, you need a forceps, the blue one above. With the help of forceps, holding it at 45 degrees angle, you place the cover slip on the specimen mounted on the glass slide. And you also need dropper when you need to place a drop of saline or a drop of KOH or drop of iodine. Okay. So wet mount or saline mount is an unstained preparation to observe live microbes. And how do you make it? Step 1 is place 2 drops of specimen on the slide. If a drop is needed, you can place a saline drop, and then you can add the specimen, and then place a cover slip by holding with the forceps. You can see at the 45 degrees angle, and then you have to observe the slide under 10x or 40x power, either under a bright field microscope, or if you want to observe under dark field, you can do so for spirochetes. So advantages of wet mount are to demonstrate the motility of bacteria by hanging drop method, example Vibrio, to demonstrate the delicate organisms like spirochetes by dark field microscope, you can use this method, and to detect the ova and cysts of parasites like entamoeba. So wet mount is a very important question. Moving on to hanging drop preparation, it is a special type of wet mount used to observe the motility of the bacteria. Here, the suspension of the microorganisms is placed on a glass slide, and then you observe it under the bright field microscope. Here the requirements are, it's the same glass slide, but you have a concave depression in the center of the glass slide. I hope you can appreciate it. There's a glass slide below with a concave depression in the center. Then you need some cover slip, you need some petroleum jelly or Vaseline, and definitely the preparation, the bacterial liquid culture, which is known as microbial suspension, and an inoculating loop. So how do you go about? You place a cover slip, you take a cover slip, apply Vaseline or petroleum jelly at the four corners of the cover slip. Then with the help of inoculating loop, you place a bacterial growth in the center. You can see in the second picture here. After which you take this glass slide with the concavity in the center, turn the concavity down and place it on the cover slip, and immediately turn the cover slip upside down. And now you can see the preparation under the 10x or 40x of the microscope. See, when you turn the glass slide upside down, what happens? Whatever drop is there on the cover slip, it hangs down. The blue drop you can see is hanging down. So that's the reason it's known as hanging drop preparation. It is used to demonstrate the motility of the bacteria. So then when you are seeing under 10x or 40x, you're focusing the edge of this drop which is hanging, and you can see the microbe, the bacteria. If they are moving, then they are motile. If they are not moving, then they are non-motile. Okay. So this is you can see there's an edge of a concave depression, and then there's an edge of the drop where you look for the motility of the bacteria. So this is also a very important question, hanging drop preparation. Next type of wet mount is a potassium hydroxide mount. It is used for detection of fungi in the clinical specimen. So why are we adding, instead of saline, why are we adding potassium hydroxide? because it digests the keratin material present in the specimens. Usually to demonstrate the fungi, the fungal specimens are the skin scrapings, the hair and the nail which are sent. So, so we have to digest the keratin present in this material in order to for the fungi to be appreciable or seen under the microscope. So here you can see the fungi after the keratin has been digested. The hyphal forms and some conidial forms can be appreciated in this picture. Next is the lactophenol cotton blue mount, which is a mounting media and a staining agent, which is used to demonstrate the fungi. Here you have three components. One is phenol, which is killing the microbe, lactic acid, which is preserving the morphology of the fungi, and cotton blue is the stain, which is staining the chitin present in the fungal cell wall so that it appears intense blue in color, as you can see in this picture. You can demonstrate variety of fungi like aspergillus, mucor, etc. Looking into iodine mount, another type of wet mount, it is particularly useful for stool samples to demonstrate the parasites like entamoeba. So you take a small amount of feces onto the slide, you take a glass slide, apply a drop of Lugol's iodine, and then you put in a small amount of feces, mix it, place a cover slip and observe under the microscope. It is particularly useful for staining, this iodine is particularly useful for staining the nuclei and the glycogen present in the protozoans of entamoeba. Then moving on to next type of specimen preparation, which is fixation and staining. So how do you prepare this? For the wet mounts, they are very temporary mounts because you're just placing a drop of specimen on the slide, placing a cover slip on it, and you're looking for the microbes. So you have to, after you appreciate or you don't appreciate the microscope, you have microbes, you have to discard the wet mount. But here in fixation and staining, the clinical material which is sent to the lab is a smear of it is prepared on a glass slide. With help of inoculating loop, you pick up the clinical sample and then smear it on a glass slide. That is, spread it in the center of the glass slide, and then you pass this after it has air dried. The clinical sample has air dried on the glass slide. You pass it through the flame, okay, to fix the smear onto the slide. That is, you want the cells, be it a bacterial cells or the normal epithelial cells or pus cells, to fix onto the slide. So for fixation, either you can heat the or pass through the slide through the heat, or you can use a chemical method wherein you add a few drops of methanol for a minute or so, and the specimen gets attached or fixed to the slide. Why you have to fix the specimen to the slide so that upon staining it doesn't get washed away. Okay, it is fixed onto the glass slide and you can observe under the microscope. So after the fixation is complete, you stain the slide so that it gives good contrast when you see under the light microscope. So such slides can be preserved for longer time. Okay, they can be used for teaching purpose. So bacterial staining methods are simple stain. There are simple stains, there are differential stains, and there are special stains. Looking into differential stains, there are again a gram stain and acid fast stains are the important differential stains you have to know. And there are some special stains to demonstrate special structures in the bacteria, like certain metachromatic granules can be demonstrated by Albert stain. The spores present in the bacteria can be demonstrated by spore stain. The capsule of the bacteria can be demonstrated by capsule stain, and there is another special staining method known as silver impregnation method to demonstrate very thin bacteria. So these are the bacterial staining methods. So looking into simple stain, the name itself is saying that you're using only single dye, single basic dye. So dyes or stains are of several types, acidic stains, basic stains and all. You don't need to know much about them, but the dyes used or the stains used in bacteriology are the basic dyes, and which are very helpful to stain the bacteria. So here you're using single basic dye to stain the specimen, which imparts the same color to all types of bacteria. And here the basic dyes used can be methylene blue, or it can be basic fuchsin, etc. And what is the use of this simple stain? It is used to demonstrate the morphology of the bacteria as well as other cells like the epithelial cells or the pus cells which come along, which are present in the specimen. So here you can demonstrate the bacteria. You can see that their morphology, that is their shape, is circular, and how they are arranged in pairs, as well as you can demonstrate some other cells like pus cells. Next is a differential staining method wherein two different stains are used to impart different colors to different bacterial cells. And why do different bacteria take up different colors or different stains? Because of the difference in their cell wall composition. So under differential staining, you have to know about Gram stain and Zeil-Nielsen stain, which are very frequently asked in exam. Looking into Gram stain, it was first developed by Christian Gram, a Danish scientist, and the Gram stain is a cornerstone for bacterial identification. So looking into the principle, the Gram stain separates bacteria into two groups based on their cell wall composition. One group of bacteria are Gram positive bacteria which stain purple in color, and the reason for them to stain purple in color is because of the thick peptidoglycan present in their cell wall. Another group of bacteria is Gram negative bacteria which stain pink in color, and they stain pink in color because of thinner layer of peptidoglycan in their cell wall and high lipid content of their cell wall. So you have to remember that Gram positive bacteria appear purple and Gram negative bacteria appear pink. So looking into the procedure of Gram stain, you take a heat fixed smear, then you pour a primary stain. You flood the smear with primary stain which is crystal violet, allow it to stay for 1 minute, then wash it off with water, followed by adding grams iodine known as mordant, which further fixes the crystal violet into the bacteria, which is kept for 1 minute. Then it is flooded away with the water, followed by decolorization step. The decolorizer used is acetone alcohol, which is just placed for 10 seconds, followed by pouring a counter stain onto the slide, which is saffronin, which is allowed to stay for 1 minute, and then it's washed away with water. And now the smear is ready to be observed under oil immersion objective. The wet mounts were observed under low power or high power objective, 10x and 40x magnification, but the stains are observed under oil immersion objective, that is 100x. So let us see what happens when you are staining. Say you have received a clinical sample where there are a group of two or more bacteria present. So first you have made a fixed smear of the clinical specimen, then you added crystal violet. So all the bacteria, irrespective of whether they're gram positive or gram negative, they take up the crystal violet. And in the second step, that is mordanting, where you apply iodine, here the crystal violet and the iodine have formed a complex and still they are, still violet in color. The next step of decolorization, where you're using acetone and alcohol, the gram positive bacteria are not decolorized, that is they retain this crystal violet color because of presence of thick peptidoglycan in the cell wall. You can see here the circular or the round bacteria are still purple in color, but the bacteria which are rod shaped, which are gram negative bacteria, have lost their uh, purple color, that is the crystal violet has been washed away because of thin peptidoglycan in the cell wall and more lipid content which are dissolved by the alcohol. The fourth step is application of counter stain, be it either saffronin or you can use dilute carbulfoxin. So you see that the circular bacteria which are gram positive still remain purple, but the rod shaped bacteria which are gram negative, which have lost their crystal violet dye, take up this pink color of the saffronin. So these pink colored bacteria are gram negative bacteria and the purple colored bacteria are gram positive bacteria. So gram positive, so interpretation is gram positive organisms appear purple. For example, gram positive cocci are round shaped bacteria, are Staphylococcus and Streptococcus. Gram-positive bacilli are rod-shaped, which you saw in the picture. They're Bacillus and Corynebacterium are the example of gram-positive bacilli. Gram-negative organisms appear pink. So gram-negative bacilli examples are E. coli, Salmonella, Vibrio, and gram-negative cocci examples are Neisseria and Moraxella. So gram stain is a very important short note. So here you can see the picture of gram-positive cocci, the circular bacteria, gram-positive bacilli, the purple colored rods. Then gram-negative cocci, the pink colored round bacteria like gonococci here, and gram-negative bacilli, which are pink colored rod-shaped bacteria. What are the uses of gram stain? First of all, they're identifying whether it's a gram-positive bacteria or gram-negative bacteria, and once this report, which is out within couple of within minutes, this helps the physician to start off the antibiotic treatment. If it's gram positive, it's a complete set of different set of antibiotics, and if it's gram negative, it's a different set of antibiotics. Even before the culture report, which takes 48 hours to be out, the gram stain, which is out within a minutes, can be useful to start the empiric antibiotic treatment. And certain fastidious organisms are difficult to grow on culture. Fastidious organisms are organisms which require very high nutrients, rich nutrient media, and anaerobes which are difficult to grow. Anaerobes are bacteria which can't grow in presence of oxygen. So in case of such bacteria, you can the gram stain helps in presumptive identification of these group of bacteria. It also helps in identification of yeast cells. Yeast cells are fungi like Candida and Cryptococcus. Next is a Zeal-Nielsen stain. It's a differential stain. It's also known as acid-fast stain. It is used to demonstrate the presence of acid-fast bacteria. There are certain group of bacteria known as acid-fast bacteria like Mycobacterium tuberculosis and Mycobacterium lepra. It is useful to demonstrate these and also demonstrate the spores. Spores are nothing but they're the dormant or the resistant forms of bacteria. Whenever the environmental conditions are unfavorable for bacteria, you know, they become spores. So you can also demonstrate the spores by Zeal-Nielsen stain. What is the principle behind the Zeal-Nielsen stain? The cell wall of these acid-fast bacteria contains a mycolic acid, a very lipid-rich uh, component known as mycolic acid, which resists the decolorization by acids and also resists the stains to some extent. Okay, this is the principle behind Zeal-Nielsen stain. So what is the procedure? You take a heat fix smear, then you flood the smear with carbelfuchsin. I told you acid-fast bacteria are even resistant to stains. So they don't take up the stain easily because they're having mycolic acid in the cell wall which is not allowing the stain to enter. So you have to apply some heat from below so that by heating the carbelfuchsin is allowed into the cell wall of the mycobacterium. So you flood the smear with carbelfuchsin and heat at intervals, not continuously. So you heat it till the steam rises from above, you heat it from below the slide till the steam rises up, and then again you have to remove the heat and repeat this for at least minimum of 5 minutes, followed by decolorization with 20% sulfuric acid for 5 minutes in case of Mycobacterium tuberculosis, and the decolorizer used is 5% sulfuric acid for Mycobacterium lepra. Okay, following the 5 minutes of decolorization, you have to add a counter stain which is methylene blue and allow it to stay for 2 minutes and then wash it out with water and observe under oil immersion objective. So there are only 3 steps here. Primary stain, which are where you are adding carbelfuchsin and heating the slide from below till the steam rises, stopping and then again heating it till the steam rises. Once the steam rises, you again stop heating. This you do for 5 minutes. Then you flood off with saline. Then you decolorize. The decolorizer used is if you're suspecting Mycobacterium tuberculosis, the decolorizer is 20% sulfuric acid, and if it's Mycobacterium lepra, the decolorizer is 5% sulfuric acid, which is kept for 5 minutes. Then you wash it off with water, followed by counter staining with methylene blue for 2 minutes, and then you observe under oil immersion objective. So interpretation of the result, if there are acid-fast bacilli, they appear red or dark pink in color. So you can see these acid-fast bacteria, which are Mycobacterium tuberculosis, and rest of all the cells, be it pus cells present in the sample, epithelial cells, they appear blue. Okay, and the bacteria which are not acid-fast, they take up the blue color of the methylene blue. So there are certain modifications of acid-fast staining known as cold method, where you don't need to heat the slide from below, like quinone stain and Gabbett stain. Why you don't need to heat? Because in one of the stains like quinone stain, the phenol concentration is increased. This is sufficient for the primary stain, which is carbylfuchsin, to enter into the bacterial cell. Okay. Next, let us look into Albert stain, a special stain which is used to demonstrate the metachromatic granules of a bacteria known as Corynebacterium diphtheria. Okay, you all must have heard about diphtheria, isn't it? It's caused by the bacteria Corynebacterium diphtheria. So these diphtheria bacteria will have certain energy granules within it. So in order to identify this bacteria, we are staining these granules, energy granules known as metachromatic granules, by using Albert stain. The reagents are toluidine blue, malachite green, and Albert's iodine. So once you stain with them and see under oil immersion, you will see the green colored bacilli. That is, can you see the green colored rods in the picture? With bluish black granules, which are the metachromatic granules, which are particularly present in Corynebacterium diphtheria. Then there's a negative staining method, also known as capsule staining, where you're appreciating the capsule of the bacteria. Here you're using India ink. Yes, you have heard right, it's just an ink, or a negrosin stain, which is mixed with the specimen. So here what you're doing is you're taking a glass slide, you're placing say the CSF sample or the sputum sample you have received, and then you're adding a drop of India ink to it, and then you're observing under low power followed by high power objective of the microscope. So what happens is the background which is there in the smear, it is staining dark. It is taking the black color of the India ink, and the organism is colorless. Why? Because it has a capsular protection around it. So it is not allowing the stain to enter and stain the bacteria. Okay. So this negative staining is particularly useful to demonstrate the capsule. Some capsulated organisms like Pneumococcus and Cryptococcus, the yeast Cryptococcus, can be appreciated by negative staining. Here you can see in this image, the background is dark in color, and you can see certain bacteria with capsule around them which are unstained, and so they remain colorless. So negative staining is to demonstrate the capsule, and some examples of capsulated bacteria, Pneumococcus, and one of the capsulated yeast is Cryptococcus. Next is the impregnation staining. I told you silver impregnation method, which is used to demonstrate bacteria which are too thin to be visible, even under the light microscope. So you apply the silver salts so that you can demonstrate certain spirochetes, and you can even demonstrate bacterial flagella by this method. So the last one is left is spore staining, which we will be discussing in under morphology of bacteria. Okay. Thank you.